Hey class, and welcome to part 3 of chapter 8. So right now we're going to talk about alterations of chromosome number and structure. So first, let's talk a little bit about karyotypes. So karyotype is a photographic inventory of an individual's chromosomes. So what it does is it lines up those chromosomes and it allows those homologous pairs to be looked at, and what it does is it stains and magnifies those chromosomes, and it does that during metaphase. So karyotypes are produced from dividing white blood cells stopped at metaphase. So what it allows you to observe is homologous chromosome pairs, chromosome number, and chromosome structure. So let's take a look at what that looks like. So here it shows the human karyotype. So we have our 22 autosomes and then our sex chromosomes. Obviously this is a male, so we have our X and our Y. So take a look here just as a refresher. This homologous pair, the sister chromatids are joined at the centromere, and it's a pair of homologous chromosomes. So that's what we have here, is our 22 pairs and then our sex chromosomes. So one of the things that you guys were asking about in class is Down syndrome. So an extra copy of chromosome 21 is called Down syndrome, and the technical name for that is trisomy 21. Tri meaning 3. So there's 3 chromosomes on that 21 spot. So it involves the inheritance of 3 copies of chromosome 21. It's the most common human chromosome abnormality. So this imbalance in chromosome number will cause Down syndrome, and this is characterized by facial features, susceptibility to disease, shortened lifespan, mental retardation, and other variations of characteristics. So this also increases with the age of the mother. So we kind of talked about in class that after the age of 35, women tend to have more problems, or they're more likely to have more problems with giving birth, and it can probably lead to trisomy 21. So here again is this human karyotype kind of blown up for you. So notice how all these homologous chromosomes are the same length, and they're dyed to kind of show the different loci. And then here's a cute little picture of a little girl with Down syndrome. So accidents that can occur during meiosis. So these accidents can change the chromosome number. So first we have non-disjunction. So it's the failure of these chromosomes or chromatids to separate during meiosis. So during meiosis 1, you guys learned about how those homologous pairs line up at the equator. So what can happen is those homologous pairs fail to separate during anaphase, and what happens is both pairs will go to one pole. And then during meiosis 2, this can happen also. So both sister chromatids, because we no longer have pairs, both sister chromatids will go to one pole. And then fertilization after this non-disjunction occurs will yield zygotes with altered numbers of chromosomes. So taking a look at a picture here, non-disjunction in meiosis 1. So we see that homologous pair not separating. And then we'll take a look at the gametes. You have that extra number of chromosomes. So instead of having the normal number, you have n plus 1. And then take a look at these other gametes. Those are now missing a chromosome, so it's symbolized by n minus 1. And then taking a look at what can happen in meiosis 2. See how these sister chromatids are pulling apart right here? Instead of this happening, both sister chromatids will go to one side of the cell. And then take a look again at the effect it will have on the gametes. So then let's talk about 8.24. So these alterations of chromosome structure can cause birth defects and cancer. So there's a couple different things that can happen. The structure change can result from breakage and rejoining of chromosome segments. So first we talk about is deletion. So it's a loss of a chromosome segment. And in the next slide it'll show some pictures and it'll be more clear. So deletion is that loss of a chromosome segment. So a big chunk of that chromosome is actually taken out. So you can imagine the kind of effects that can have. Duplication is the repeat of a chromosome segment. So instead of being deleted, it's rather duplicated. So you have extra amount of DNA information. Inversion is the reversal of a chromosome segment. So genes that are side by side on different loci might flip-flop and reverse inside the chromosome. Then translocation. This is probably the hardest one to understand. Translocation is when two non-homologous chromosomes will experience crossing over. So remember, when we talk about crossing over in prophase 1, it's always with those homologous chromosomes. So in translocation, they're non-homologous. So they are not supposed to be exchanging genetic information. So translocation it can be a really, really bad defect. So altered chromosomes carried by gametes can cause birth defects, and if this happens in somatic cells, normal body cells, it can lead to cancer. So then let's take a look at some pictures here. So deletion, see that piece of that chromosome is taken out and it's shorter here. So you can notice the effect. Then with duplication, that piece of that chromosome duplicates, and you can tell that chromosome is longer, so you have more genetic information than what you really need. Inversion, that is when pieces of genetic information inside the same chromosome is flip-flopped. So you can tell by the colors here that those have reversed. Then reciprocal translocation, which is the hardest, I feel, to understand. These non-homologous chromosomes, notice how they're different in length, so that's the key thing to let you know they're not homologous. These will exchange DNA, and then look here at the result. You have two chromosomes that have exchanged genetic information. So now that we've reached the end of chapter 8, there's some things that you should be able to do. So make sure you can identify the roles of cell division in living organisms. You need to be able to distinguish between the events in interphase, mitosis, and cytokinesis. Describe the movements of chromosomes in all those stages. Define the following terms, which there are a lot there, a lot of those C terms that you have to make sure that you do not confuse. So, be able to compare and contrast mitosis and meiosis. This is really big. We've talked about this quite a bit in class already, and you did a worksheet for homework, hopefully, that really stressed the importance of that. And make sure you can distinguish between the terms in the following groups, such as haploid and diploid, so N, 2N, sister chromatids, non-sister chromatids, deletion, duplication, inversion, and translocation. You also need to be able to describe how genetic variability is generated through meiosis. So we talked about random fertilization, crossing over, and how those chromosomes or sister chromatids align at the equator. So you also need to identify factors that control cell division and describe how cancer cells escape these controls, which we also talked a lot about, those cancer cells not obeying the normal laws. So I hope you enjoyed this first screencast done by Miss Williams, and I hope you have a good evening.